Hello, everybody. Welcome back. So as I think a few of you are seeing here, I've got a poll up. So you can go ahead and answer the poll. Just want to gauge where everyone is kind of at right now in terms of their preparedness for the afternoon workshop. I know that it's a little more complex than some other more simple demonstrations. Unfortunately, it's pretty necessary. It's an odd challenge because doing this type of research is difficult. Things don't always work. Things don't always work when you're even in a very well-supported kind of high-performance computing environment. Troubleshooting is always a big part of it. And one of the goals of this whole week really is to also get you thinking about containerized computing, how to use Dockers, how to how to work with GitHub repos to access data and all of that. So let's see. So I'll also encourage you for the afternoon session, since I won't be just kind of talking nonstop and delivering slides as in the morning. Definitely feel free to ask plenty of questions, chat here. I think Aaron was saying the Riot rooms are up for chatting with TAs. The GatherTown is open also for troubleshooting and chatting with TAs and chatting with each other. I definitely encourage you all to work together. Hajer has also been helping out. She'll be a TA, I think, for day 5. She's an honorary TA. Looks like she's got our studio working on Signet. So for those of you who need to work around having an issue with Docker Desktop or unable to get Docker working locally, she may have the solution that we're all looking for. And this is good. So far, we've got one person who's troubleshooting. It seems that a lot of people have Docker working, have not yet cloned the Git repo. Okay. So, and there's a few people who seem to have it all up and going. So one of the nice benefits of the Crowdcast platform is that we can, obviously this can be revisited at any point. Everything is, set up to be reproducible. The GitHub repository will be available for perpetuity and continually updated throughout the week as we build the workshops. And the data that we've made available for download to work on for some of the larger data sets, like the genetic data set, which I've hosted on Figshare, and we have code in the R notebook, which is inside of the Git repo, that gives just the one-liner of how to pull that data down into your local machine and into the GitHub mounted volume. So I'm going to share again. I'll share my screen. I'll go over a few of the kind of what it looks like on my machine. I'm using a Windows machine, a ThinkPad with Windows 10 with Docker Desktop, and this actually looks quite good. We've got quite a few people who have answered the poll. 16 out of 40 people have answered the poll. I think some people are afraid to say that their computer is their enemy. And unfortunately, in neuroinformatics, it is the key. The computer is the key. Well, your brain is the key. The computer is a necessary tool. Reshow cloning from inside RStudio. Yes, so what I'm going to do is I'm going to kind of go from from the start, since it looks like most people who are answering the polls have it working, they're actually inside our studio and they've cloned the Git repo. I will show you what it looks like on my computer. If you're unable to get it to function that way, then we'll have to interact with some of the TAs. Again, if we pull ahead, if you still aren't able to get to the stage that we're at, kind of in the moment, don't worry too much about it. I'm going to have a kind of fair amount of time left open for Q&A, for some interactions. If anyone who is kind of in the audience or a TA wants to also come up onto the stage, and I can give them video if they've been tackling a particular issue and want to show how they've solved that issue on a different operating system or maybe on Cynet, then we can take time to do that as well. The code has mostly been designed so that it runs quite quickly. And if the environment is set up correctly, then we shouldn't have too many problems. But generally, just getting your computing environment arranged, getting the data where it needs to be, and having everything set up is the most time-consuming part of most analyses. It's really at the core of everything that we're doing, and a lot of the key steps that we're actually going to cover today inside of the environment using PLINK and looking at the genetic data that's available for download is all about data management and making sure that everything is in the right place. So given that it is the first day and we're expecting to run into these problems, don't panic if we kind of pull ahead a little bit. We'll have time as well in the afternoon break and some kind of more downtime so you can troubleshoot your issues. Better to get it out of the way now and get more comfortable with some of these techniques and processes for the rest of the week as well, because you can always come back and revisit this day's material, and all the code will be there. So I'm just going to go ahead and share my screen. It's encouraging that no one's given up hope. I think that's wonderful. You can't give up hope. It's all possible. It's very possible. And thank you very much to everyone who is texting and the TAs who are helping out and troubleshooting with everyone. It's absolutely vital, especially in this very challenging environment where we can't really see each other and talk to each other normally. So we're definitely having some more issues. And that's right. Yeah, Aaron says you can always pause me and watch it later as well. So don't worry about that. The idea is if you're ready to go, if you're here, if you're at this stage, we can go through the material. I want to make sure that we do get through the code so that when you do finally get your environment up, you know what you're aiming for and what the expected outcomes are. So what you can see here is I've got open a PowerShell that I'm running as administrator. And you can see I have my Docker run command. I apologize if it's small. I'm going to try to make it bigger so that everyone can see. I have my Docker run command exactly as it is on the GitHub. I have my own local directory that I've created in KCNI Summer School. I'm actually in a test directory right now, which was where I was testing, pulling everything in together. The mount that I've created. So my local folder is called test. It was just an empty folder, and I've essentially mounted this to this KCNI school data directory, which exists inside of the Docker. So you may remember from some of the earlier slides in day one, this kind of virtual environment is going to be called this inside of home, RStudio, KCNI school data. That exists virtually inside of Docker, whereas this is a real location, the C drive inside of my documents, a test directory, a real one. And then there is the latest version of the Docker image that's been started up. So if you do run it and it's running correctly, it should look something like this. It just tells you it's looking for files and everything's been completed. It seems that most people are at the stage of either having Docker, kind of considering the poll, are at Docker and either have or don't have the repo installed or cloned. So once this is running on your computer, you need to enter the web browser. This is the location that I've set, localhost 8787. And it opens up a virtual RStudio environment. Now, you won't necessarily have anything. When you open up the first time, you should see something like this. Just a blank console, which is the R console environment where you enter code. The source code, which is where we can open the R notebook or file, won't be there yet because this will essentially be an empty directory. So right back to the start, this is what you will see. At the bottom here in the RStudio environment, we have three options: console, terminal, and jobs. We're going to make use of both the terminal and the console for this run through. The terminal essentially creates a bash scripting environment. It's the same type of terminal that you would get if you had just a Linux-based machine or like a Unix machine, like in Apple computers. The Docker run command, again, so I can show you how I've mounted in this location the virtual environment. So here's my C drive users, Felsky documents, KCNI summer school test. But inside the Docker environment, this location is a home, inside of home directory, RStudio, KCNI school data. So this is where there's the interface between my real computer and the Docker environment, is this folder, test and KCNI school data. They are essentially now the same thing. So here it's created this KCNI school data folder. And what you now want to do is essentially work inside of that folder. So that folder is mounted to the folder. Right, we're getting requests to slow down a bit as you go through things. Right, we can go back again. If Docker is functioning correctly, and if you've run the Docker pull command, which means that this Docker image exists on the computer. So this code is on the KCNI school environment website. So we can go here to the KCNI school lessons website. This is the GitHub site for Krembil Neuroinformatics KCNI school lessons. And inside of in envs, which is where the environments are, there should be a link to this as well, is the setup for Docker. Now again, going by the polls, I have two people who are currently troubleshooting with TAs. It seems that most people seem to have Docker working, so I can only really go on what I see here. So I won't belabor getting the Docker to work. Of course, there's a lot of reasons why it may not work, and it will help out. And we have Isabel seems to be posting something that may help with troubleshooting some errors. So the two steps that are important to note is Docker pull and Docker run. Now these are separate steps. Let me zoom in a bit here. Right, Docker. This single command here should install the Docker image on your computer. That's one step. It's one potential way in which things may not work. At this point, you have to have Docker Desktop installed. Docker Desktop is just the program that allows you to pull in images and run them. So this is something that you really only have to do once. So I am running in PowerShell. This is Windows PowerShell. And I'm running it as an administrator. And these commands all work inside of Windows PowerShell. Daniel's asking about running the command, nothing is happening. So if you could, so it looks like there may be issues with depending on your operating system and depending on whether you're in a Mac or Windows, how you specify your local file location in terms of backslashes, colons. Windows does it differently than Mac. This is a Windows machine. So there's going to be some discussions in Riot about how to get this to work. So again, I think in terms of technical issues, just getting Docker to work, I think thank you, Sunny, who's coming in to check. Let's see. So I'm gonna continue, just again for the sake of individual troubleshooting. Please continue to interact with the TAs and the other members. But it seems that unless you're not really there yet and you haven't responded to the poll, please go ahead and respond. It's the only way that I can really get a sense for the larger group and how many people are kind of ready to proceed to the next step. But we have to keep things moving, and again, it's all being recorded. So the step that it seems we need to start looking at is once the Docker is up and running, once we can actually start in RStudio, we're going to clone the Git repo. Now, some people, a fair amount, have already done that. Some have not. This is, again, once it's been fired up, and depending on how you've pulled your, started your Docker environment, this file structure, and the example, I believe, is inside RStudio KCNI school data. So this just means that whatever file you have locally on your machine, or whatever your directory is that you've chosen to mount onto, it will still be called inside the Docker environment this. This is what your directory structure will look like inside the virtual Docker. So we can see that in the terminal of the RStudio. This is a bash shell. It uses bash scripting language. We can look at files. We can move around. ls is the list command. It shows you what is in the current directory where you are. pwd shows us the current working directory. It's print working directory. So you can see that we are inside of root, which is the first slash, home, RStudio. That is the virtual file structure that we've created for our Docker that actually exists in real life on our computer inside this test folder. So this is the interface between the real stuff and data that we will download and clone on our computer. When we do that inside the Docker environment, all of those files and everything we do will appear inside this test directory. So we'll be able to see it on our local machine, and we're operating it inside the environment inside of this folder. So what we need to do is enter that folder. We do ls again, we can see there's KCNI school data. So we want to be inside of KCNI school data. So we cd to KCNI school data, ls. Now you'll see I have here a directory called KCNI school lessons. This is because I've already done the git clone of KCNI school lessons. I did mine, I believe, in Sidwin. I think I will just go and try to do it again right here, right now. So here is the KCNI school lessons GitHub site. This is the location. All you need is the URL for this repo. This URL is specific to this repo. It contains code and some data for each of the day's workshops that we're going to do. It can be removed and recloned. It can be updated every time that we add new data to it. As it stands right now, everything should be in place for day one that we'll need. So when we clone this, it will create that folder. Go back into my, oops. Go back to my RStudio. We use the command git clone. So in this case, since I've already done it, I don't know if it's going to create an error or if it's going to create a second directory on it. But what I'm going to do, just for the sake of demonstration, KCNI school lessons. Let's just run it. Git Clone. And Git Clone will create, yeah, the webcam is a good point. I don't actually know if I can move it. What I can do is I can reshape my RStudio window so that it's not obscured. Is that a bit better? Yeah, especially as we get into working with the terminal. So Git Clone, we're entering in terminal. So remember, when we're in console, we're inside of R. We're running R code. When we're in terminal, we're in the environment around R. We're actually inside of the Docker using a bash terminal that allows us to navigate and use other types of softwares that don't exist inside of R. And we'll be doing that with 2 different software packages today. So let's try the Git Clone. In order to paste text into terminal, you can't right-click and do it, which is frustrating. You can paste from any source into here with, on Windows machine, Shift Insert. On Mac, I think you can do Command Insert. To be honest, I'm not 100% certain how it works with Mac, but it's something along those lines. For the for our, I think it should be for Windows machine, Shift and Insert should work for everyone. And when you run this Git Clone, it's telling me that I have a fatal problem because it already exists and it's not an empty directory. So this is my, I've already done this. So I did this beforehand. What I'll do is I'll make a new directory called, new. I'll call it example. This would just be our KCNI school data. But running that same code, this Git Clone, and then the actual GitHub location will reach out to GitHub and should access it and recreate the entire file directory wherever we were located, right there. So you can see it's receiving objects, it's downloading at a decent pace, although I wonder how big it is now. So we've put every day onto here, but this is what you should be seeing essentially. And the important thing to remember is that when you're cloning this, when you're running this clone command to pull this KCNI school lessons folder from GitHub into your Docker environment, you want to make sure that you're in the Docker directory that you created when you ran Docker run. So that virtual mounted folder that we called KCNI school data is where you want to be, because it means that now when we work on files and we create files and we perform analyses inside of KCNI school data, which now will contain this new cloned KCNI school lessons, those files will exist and they'll be saved in our directory on our local computer. They'll be mounted locally. So that means that we can go back to them. We can shut down the Docker image. We can kill the Docker image and then run it again another time. The files will still be there. So it's going pretty good. Checking out files, done. So that took maybe 1.5 minutes, 2 minutes to download. Are there any issues? Is anyone having continuing to have problems with Git Clone? Any kind of specific that they're not already dealing with, with in Riot or in the GatherTown? Is there any kind of pressing questions related to the Git Clone? So as you'll see, I'm still in the terminal. I will ls again. And now it shows me it's created KCNI school lessons. Now I've done it inside of this folder called example, which is still inside of KCNI school data, which is actually still not getting the cloned data inside RStudio. So it's specific to that. Is there an error that you're getting when you're running the code? Is there something specific? Maybe try to interact with some of the TAs to figure out exactly why that clone command isn't working. So that's wonderful. Thank you, Buzma. So, okay, people are in Gather and Riot. So everyone's digitally mingling. It's wonderful. So it's important to be able to clone this repo, because this is where we've put the code, the R notebook itself for day one, and then also we will have a lot of other intermediate files, so that the idea being that not all of the code will have to work perfectly for everyone. We have different computers. We may even run into some problems today when we're running some of the more demanding software, but by keeping everything on GitHub at any point and after the summer school is complete, you'll be able to go back and try these steps again with the final data, and actually even look at files that were generated from commands that you might not be able to run on, a Microsoft Surface or a very small computer. Again, we are attempting to recreate some of the types of analyses that you would work with in this kind of high-dimensional neuroinformatics space, which is not, as I said initially, not an easy thing to do necessarily. So these are natural growing pains, and if you're having issues, continuing issues with Git and with Docker and just dealing with the virtual environments, don't worry about it. It's in what we're going to cover in 5 days. You should be feeling frustrated. You should be feeling that you have something to figure out, and you'll get there. And we're here to help you get there. So even if that's all that we accomplish by the end of the week, is some ability to operate inside of a virtual environment like this and pull code and get some of it to work, we've, I think, done our jobs. So what I'm going to do is I'm just now going to remove this example folder that just showed the command. I have to move RF just to keep things clean. And this is where you should be, or where you will be, once you can get the Git clone to work. And once that happens, it's just taking a second to delete all of those files. Once that happens, this is what you'll see. Hopefully, we should be, if you run pwd, you should be inside of the mounted folder, the digital folder that you've created that resides at that location that you've mounted to on your computer, KCNI School Data. And in this folder now, we have the cloned KCNI School Lessons directory. So let's go in there. Let's cd into this directory and look at what we have. And you can see it's downloaded all of those folders that you see on GitHub. That's what it is. It's a clone of this file structure. Everything that's in here is now on your computer, and it's operatable inside of the digital environment. We want to go inside of day one. We can change directory into day one, and you can take a look at what there is. What we have here is a few files and a couple directories. Now, you're not going to see these directories, Pheno, PRS, and Geno. You know what I'll do is I will actually remove those and just go step by step with you with what you will see, so that there's no confusion as to what you should be expecting versus what I have here on the screen. So if we look at what we have here, we have an HTML document, there's an ND HTML document, and an RMD file, as well as a README for MD. Now the RMD file stands for R Markdown. It's the programming language that we've written, or at least for this section for R, that we've written the tutorial in. It has some nice features. You can put in chunks and blocks of code that are functionally related to each other. You can include coding languages other than R inside of the R document, such as bash code, which we'll be using for the first bit of the tutorial. You can highlight and describe the code and perform much more detailed annotations, and it's also much prettier. And that is what you'll see in the HTML doc, which you can open up in a browser. You can also print to PDF and make very nice documents to preserve the code. As Aaron said at the very beginning this morning, about your biggest collaborators yourself 6 months ago or 6 years ago. If you've written a nicely annotated code and you understand where everything's coming from, you're going to thank yourself many, many times down the line. So what I now encourage you to do is to open this R notebook inside of R. So it will pop up in this top. I've already opened it. The way that you can do it is you click this Open Existing File button. You can also go File Open. Now, because this is a virtual R Studio environment, these folders are not going to be folders that look like they're on your computer, right? We're not in C, Users, D, Felsky, Home, anything like this. Remember, we're inside the virtual environment. We're in Home, R Studio, KCNI School Data. There's the KCNI School Data. And look, sure enough, there is the Git repository that we've just cloned. So we can go inside of there. We can go inside of day one. And we can open our day one code, our Markdown file. And when we open that, this is what we get. So I'm curious now, if you're with me so far, if you're kind of here, can we do like +1s? I feel like there should be a generic polling option or something, kind of like a hands up, hands down. I guess I could do a poll. I could do a new poll. I can create a new poll. So I'm creating another question, and it's, are you with me? Answer 1 is yes. Answer 2 is no. Answer 3 is in spirit. We've got some +1s. That's good. I like +1s. We've got the great. This is good. It's okay to answer no. Remember, this is a big part of why we're here. This is why we're doing it. We know this is going to be a grind for some people to make sure that we can get functioning, but we've got a lot of +1s, which I like. This is great. In spirit, thank you. That's wonderful. It's good to be in spirit. Good to be there emotionally. This is good. Good. We've got a couple no's. Excellent. Some are with, some are not. We're working. The balance is shifting toward no, but we've got lots of +1s. Okay. This is okay. This is looking good. This distribution is looking good. 2 in spirit. So. So the majority appear to be yes with me. Some in spirit. I got 2. Some people are given +2s now. This is great. So I'm going to go to the next step. The next step is not a computational step. There is one more thing that we have to do to prepare ourselves for today's tutorial, and it's a download. It's not a GitHub repo clone. It is just a direct download from a data hosting website called Figshare, which allows you to freely host data sets, figures, supplementary information, anything that pertains potentially to things like workshops or publications. And it's a big kind of hub for open science to facilitate that. If you've gotten here and you said yes, and you have the R Markdown notebook open, I've outlined a couple of the basic goals of what we're going to do along the lines of what I was speaking about before the lunch break. And then here in step 0 is where we begin to just make sure that we're oriented in the right directory, that we've gone into the KCNI School Lessons Day 1 directory that we got from Git Clone, and then to download the Figshare data. And that's accomplished with a single command called wget, which just pulls data from a given URL that's hosted here and will create a folder, a zipped folder called Day 1 Workdir. So the first piece of this code, we can now copy and paste directly from the R notebook and put it into the terminal. And again, to copy and paste directly in there, we can copy. Now this is bash code, remember, so we can't use the kind of chunk syntax because it will attempt to run that code inside of R into the R console. Instead, we're navigating through the terminal outside of R, and we'll copy this command, which is just change directory into KCNI School Lessons Day 1. Now, I've already navigated in here, right? You can, at any point, remember, PWD will show you where you are in your file structure. We are all the way in Home, RStudio, KCNI data, KCNI school lessons day 1. That's where we want to be. If you are not there, then you want to use CD for change directory and get into KCNI school lessons day 1. So you may have to replace this day 1 with the full path of where you want to be. For example, because of the way that I've done my mount for my Docker, and that I remember I created it called home RStudio KCNI school data, right? That was the mount in my command, right? In my Docker run, home RStudio KCNI data. So that's where RStudio thinks I am. Home RStudio KCNI school data, KCNI school lessons, which I just downloaded day 1. So that's where we want to go. And then we simply run this wget command, which I will run by doing shift insert, put it there, press enter. And hopefully, with some luck, this will be happening. Now, interestingly, it's going much slower than, well, perhaps not interestingly, or not surprisingly. It's going much slower than, it's not a ton of data. It's about 400 megabytes. It's smaller than the Docker image, quite substantially smaller. It's 450 megabytes of zipped data. So I suspect this will start to speed up. For some of you, it may be a lot faster. For some of you, it may even be slower. So I recognize that. It's true. I could, because of this broadcast, I could temporarily disable my video and see if that boosts things. So I'm going to toggle video off for 1 second and see if that changes download speed. It has substantially. So maybe it's going faster for everyone else who isn't doing the live broadcast. So I'll keep the video off just for a minute to help with the download speed. But definitely, because I'm hosting the crowdcast and also sharing my screen, there's a lot more data being transmitted through my connection. But this is okay, given the, I think, we still have, from looking at this poll, there's still almost equal between no and in spirit and yes for being with me. So yes, it's a good suggestion, Sri Joy. So we have some time again for more questions or answers while this downloads. As for RStudio and using RStudio notebooks, there are a few tricks and things that I'd like to bring your attention to in case you're not a very experienced R user. The RStudio interface of this multi-paneled window is useful not just for running R, as I said, but also has this very handy terminal function. Each window can be adjusted for its size, and I'll show you what's in each window in a second. The console, again, is R code. Everything that comes into console is R code, so you enter functions and direct blocks of R code into this window, and then again, terminal is a bash environment. Actually, when you first start it up, it won't be bash shell, born again shell. I think it'll just be shell. You'll want to type in bash, but that will be in the code. We'll actually do that in a second. In terms of running R notebooks, there are some options. Any bit of code that is surrounded by the 3 ticks, we can tell the R notebook what type of code it is. In this case, it's shell code. When we create a final document, there are some features of R notebooks that will generate things like figures and outputs from the code and store it inside the script itself, which is different than a regular R script, which is just code. It's just alphanumeric text. In this case, in the R notebook, we can actually store more rich data types. In this case, I've put the function as eval equals false. And that's because we want to physically copy and paste this into terminal. We won't be running it in console because it is not our code. However, as we scroll down, we'll find most bash. A lot of this is going to be, again, working with the genotype data files and the summary statistics files that we're downloading now. And keep in mind, for the purposes of this workshop, we're using real data. It's not minimized toy data that doesn't actually look like real data. I think that's really important because often, especially sometimes, actually PLINK toy data sets are a complete joke. They look virtually nothing like what you'd encounter in a real analysis of this kind. It also gives you a sense of the scale of the types of analyses that are being done and some of the requirements of the code and processing in order to handle large files. And part of this is learning the tools that have been developed to read very large files. So most of this will be done in Bash up until we can scroll all the way down. And also if you've already downloaded the data, if you want to jump ahead, obviously you're welcome to go at your own pace. If you're just flying along, you can try things out. Everything should be written in this case. Everything's written out. The code should work in a self-contained environment. So feel free to jump ahead if you are getting ahead. And if it's just too slow for you, or if we're getting hung up on things, and we can have a chance for some of the others to catch up during this time. One of the nice things about R notebooks is that when we are operating with R code, the highlighted area of the notebook is one chunk that has been kind of denoted by those ticks again and the R code. And in this case, I won't run this chunk right now because much of what the files that are being read and the operations that are being done depend on files that we're going to generate above using PLINK and using the precise software. But you have the option to press this little play button that says run current chunk. This will run all of the code in this block and output whatever outputs come from it. If there are graphical elements or text outputs from the code, it will actually put it right below this chunk inside of the R notebook itself. So you can always see what's been done if you go back through your R notebook. Once you've completed writing all of your code in the R notebook, you can do something called knit. This is what we did to generate the HTML file. That simply runs everything in your notebook. It goes through it and runs every block of code unless I've set eval equals false. Because remember, we won't be able to evaluate this code inside of our console. We enter it manually into the terminal. But it will go through and then run all of the R code down below in chunks. And it will spit out all of the outputs below and the plots and everything that we generate in R, and then save it to an HTML file, or if we knit to PDF, it will save it to a PDF file. And that's a nice portable way of sharing the notebook with other people, with collaborators, with students. It's just a nice kind of a nice friendly file format to work with. So my download is completed. Took a bit longer than expected. It's 6 minutes and 10 seconds. But now we're back here into the terminal inside of day one where I've downloaded it, ILS, and I can see that day one Workdir is here. So here we are in the notebook. We've just completed the wget to download the Figshare. Now we can orient ourselves in various ways. PWD, LS, remember print working directory, show us what's in the directory with LS. These are just the tools of the trade. You should be very comfortable using these to navigate directories inside of any bash environment. The next thing that we have to do is just set up that Workdir. So it's been compiled and stuck together so that it's not an enormous file to download. But some of the files themselves that need to be operated on are quite larger. They've just been compressed. So we need to unzip those files, and we can do that simply with these codes inside of the bash environment. So we ensure that we are in the correct directory, right? We're inside of day one. This is where the zip file that we just downloaded is located. And I will copy and paste the unzip command. And you can see it's extracting a set of files from this working directory. It's been set up. The next bit of code in this chunk, these unzip and make dir commands, all just serve to set up the day one folder. So now instead of cloning a Git repository with several gigabytes of data inside of it, we clone a very small repository that has things set up more or less the way we need them to. And then from the second source, from Figshare, we bring in the kind of meat of the data and then set things up for our analysis. So as long as you're inside of the day one directory, these codes will accomplish what we want to accomplish to set this directory up. We now look at what that unzip has done. It's just created the Workdir folder. And if we ls the Workdir folder, we can ls to see what's inside Workdir. You'll see there's a few directories. Now, I've set these directories up. One is code, which contains some code. One is geno.zip. This is where all the raw genotype data that we're going to be using is located. And it's been zipped, again, to preserve space for download. A pheno directory, which contains some phenotype information, including some scales of depression, some scales of synaptic markers in the brain. A PRS directory that we'll use to output files from when we calculate our polygenic scores, and then a zipped folder that contains the summary statistics that we're going to use to calculate our scores. And these are summary statistics that are downloaded from the consortium that performed that major depression GWAS led by Howard. So again, we are inside of day one. This code has been written. Sure, Srijoy suggests that I look at the files plane. I don't use it so much, but you may find it helpful, especially if you are still getting acquainted with navigating inside of a RStudio background environment. So this pane gives you kind of like a Windows-style view of the folders in the directory that we've created inside of our virtual environment. So you can navigate here and click through folders as though you just had an open Windows or Linux or Mac kind of user interface. And here you can see those files that you've downloaded and this Workdir that we've just now unzipped. So it shows you where you are in the file structure. It shows you what files are there, and is kind of a nice complement for all of that together. I'll quickly note up here is another pane that gives you code history, environment, connections, depending on if you're using Git or other version control and projects, which we won't be in this case. I have some items in my R environment. We'll talk about this when we start operating on the R code. But for now, we're still in setup mode. We've downloaded the Workdir. We've performed the first unzip. Now we're going to unzip the remaining files that came with it, the geno and the sum stats, and we're going to create inside of day one the file structure that we want, right? So here we are inside of day one. I'll go to the pane on the right. Right now we have these codes, but we want in here folders for genotype data, for sum stats, for PRS, for our output. And we want to make sure that everything is unpacked and unzipped and ready to be analyzed in there. So that's all that this code does, right? This is an unzip call. We're creating a directory inside of day one called geno, and we are unzipping the zipped geno and sticking it into that directory. So if I run this code, copy, shift, insert, it's now inflating the genotype file. And you'll see what it is it's telling us the names of the files that are coming out of this zipped archive. In this case, the first file coming out is geno_ro8_n1000.bed. And this is important. I've named the file according to some initial quality control steps that I've performed post-genetic imputation. So the subjects in this data set were genotyped using the Affymetrix 6.0 platform, a 600,000 SNP genotype platform. And then I imputed these genotypes, 600,000 of them, against a reference panel, the latest reference panel from the TopMed reference consortium, which is an enormous reference panel with several, it's actually hundreds of millions of genetic variants that they've identified across tens of thousands of people. In fact, it'll be over 100,000 people soon. The fact that they identified genetic variation among their sample does not mean that they are all SNPs that we can use in our analysis, because again, some of the variants are not actually present in a majority of people. They can be quite rare. In addition, during the imputation, there may be a lot of genetic variants of these hundreds of millions of variants that were not imputed with high confidence. We talked a little bit about that. There may be a very poor ability of the algorithm to actually decide if a given individual is homozygous or heterozygous at an unobserved genotype. And so every imputation procedure generates quality control metrics. They generate a imputation quality score, which basically says this genotype has a certain amount of confidence that it's imputed correctly. And a lot of times that is proportional to that kind of dosage. When we look at dosage that is assigned to an imputed SNP, if the values are all very close to 0, 1, and 2, and there are very few people in between at some point, then you might have a SNP that's imputed with a higher confidence than one that is kind of getting estimates of genotype membership that are all over the place. So the initial pruning that I've done from a data set that is over 16 gigabytes for 1,000 people is I've brought it down to a much smaller, manageable size with a certain number of SNPs. And when we run PLINK, I'll show you how many SNPs we have. The sample size is 1,000. That's why I have n equals 1,000. And I've taken out all of the SNPs from the data set that had an imputation quality of less than 0.8, the number ranging between 0 and 1. 0.8 is a kind of standard threshold that works to ensure that variants that we're actually using are imputed with high quality. And that's very important when we're performing any downstream analysis, because we want to make sure that when we're generating a polygenic score, we're not miscounting alleles that could actually contribute quite strongly to genetic risk. And you'll notice it inflated two other files. One is called BIM and one is called FAM. So this is how genetic data are stored most commonly in what's called the PLINK binary 1 format. It's been around for quite some time. It was the standard for storage and analysis of genetic data for a long time. The BED file stands for binary PED file or binary pedigree file. It actually contains the genotypes themselves. It contains a list of subjects, it contains a list of SNPs, and then in each matrix, which could be enormous. So remember, if we have 5 million SNPs and 1,000 people, it's going to have 5 billion individual cells in that data set. And each one of those cells will have 2 pieces of data, right? GA, AG. Or if it's been stored in a certain way, it can be stored as a dosage, as a 0, 1, or 2. The BED format takes genotypes only and compresses it into binary format. So you can't actually read this binary format. It is not a CSV file or just a plain text file that we can look at and see genotypes. It's been compressed so that it can be read faster and operated on faster, and it's what really makes working with this type of data possible. The BIM file, which we'll look at in a second after we've finished doing some of the other unzipping, is a list of all of the data for each SNP. So we want to know what is the ID of the SNP, right? What's the RS number of the SNP? Where is it located? What are the alleles that it has? What are the minor alleles in that? It's basically annotation data for the SNP data. And then finally, the FAM file is the same, but not for SNPs, it's for the subjects. What are the subject IDs? Are they male or female? Is there a linked case control status? The file format was actually originally designed to work with family level data, so you can store information about their parents. What are their parent IDs? Are they also in the data set? This kind of thing. So we'll look at the file structure in a moment. First, I'm going to copy and paste this extraction. Again, this is just pulling this code directly from the Markdown file that you will all have. Again, creating this directory and unzipping the summary stats and putting it into that directory. And I'll grab the rest of this code, which is, again, making a phenotype directory, making a PRS directory, and then I'm going to remove the zipped file that we downloaded and the unpacked Workdir, which is now actually just extra information. So after we've done that setup, now it looks like what my directory looked like in the first place. I have these Geno, SumStats, Pheno, and PRS folders. And these are the folders that now contain the data that we'll use to perform our polygenic analysis. So we move down, we'll get to the first steps of what we need to do to calculate our polygenic score. We're going to use a software package called Precise, and it's Precise version 2. I have the URL here, the link to the website, which is full of excellent documentation, the history of versions and updates, good quick start guides, and good detailed guides to every function that is used. And it's actually quite well documented. There's also a forum, and the authors, the individuals who work on this and maintain the program are very good at responding to comments and questions. We fixed a number of bugs just by interacting with them through the forum. Here are the things we need to do. Here are the key things. We need two things, really, to calculate the score, as we discussed before lunch. We need a set of genotype data for a target sample, right? Preferably, we want imputed genotypes. The more genotypes that we have, the more points of variation that we've accessed on the genome, the more chance that there is we'll be able to match our data set to the summary statistics, right? Not every single person who performs a GWAS and not every group is using the same reference data. They've had to quality control their data. They've removed SNPs. They've done a lot of different SNPs in order to make sure that we can match our list of variants directly with variants from another person's sample. Imputed genotypes are the way that we want to go. Remember that some of the earlier platforms and still some of the more common platforms will only actually genotype using those oligonucleotides, actually directly observe genotypes at between 500,000, maybe a little over 1 million locations in the genome. And yet the most recent analysis of TopMed found about 800 million sites of single nucleotide variation. And that, again, that includes very rare variants. If we look at more common variants and just get rid of all those really rare variants, we're still talking between 10, 40, even 80 million variants, depending on the number of people that you're looking at in your sample. So there's a lot more variation out there than we can actually reliably or cheaply measure directly. So imputation is actually absolutely necessary to fill in the gaps that are left by a traditional genotyping experiment. Because the actual biological function of a given SNP may be unknown or yet to be discovered, and because we do not necessarily design the chip arrays used for genotyping around whether or not a SNP is a functional SNP, whether the SNP itself is a known risk SNP, although there are custom arrays and sometimes this is done, we need to maximize our search space through imputation to make sure that when we are running our analyses, A, we can match it to other people's data, right? If we impute all to the same reference data set, then at least we know we'll be looking across the same set of sites. And B, we can also observe associations with imputed variants that we haven't directly genotyped that may be actually very important functionally and structurally. The way that we access our genotype data and the way that it's stored, I've just gone over the BED, BIM, and FAM files. We'll look at it in a second. These are the PLINK file format data. They are compact, portable. They're very commonly used, although they do have limitations. The second thing that we need for our PRS is the summary statistics. We need data from a genome-wide association study. We can choose this genome-wide association study. The only reason why we are doing this on major depression is as an example case. You are not limited in any sense by the GWAS choice of outcome phenotype. You can generate a polygenic score for anything that has GWAS stats available, any set of genome-wide summary statistics. And as we discussed this, they look like they are just a text file with per SNP summary stats. What is the name of the SNP? What is the effect allele of the SNP? And what is the effect size of that allele, right? What's the beta coefficient or the odds ratio? What is the standard error? At the most basic level, all you really need to know to calculate this kind of initial polygenic score, this kind of elementary polygenic score, is the SNP name, the allele that matches the effect allele, and then the effect size of that allele, that beta coefficient or that odds ratio. So to start processing the genotype data, even though it is fairly high quality data, we've removed, remember, all variants that were not imputed with very high quality. We've also removed some variants that are extremely rare. We still need to perform a set of quality control steps before we can calculate our polygenic score. And I'll take you through those now. So here I have the bash command, which just tells the terminal that we're going to be using bash syntax. That actually helps because I think sometimes PLINK will fail if you haven't started a bash environment. I've already done so, so I would encourage you to just run bash to let the RStudio terminal know that we'll be using bash syntax code. You can check to make sure that PLINK has been installed on the Docker image, should be embedded in there, it should be available. You can just check quickly by pasting in this command, PLINK -- health. And you should get a whole bunch of information from PLINK telling you the options that it has. There's a lot of options if it's installed correctly. Now, if you get nothing, if it says cannot find PLINK, Aaron has created a kind of fix-it script. It's a bash script, this .sh script. You can source this script inside of the terminal, and I think it might fix some of the path variables. It may bring PLINK back to the forefront. So if for some reason you run PLINK help and you get nothing, you can do this source command, and that may be able to fix your issue. If it doesn't, again, we got the riot rooms and the gather rooms. So we verify that PLINK is in fact installed. It is here. So now we go through quality control and data handling for the PLINK files. I've outlined how these files are encoded. A great website is the PLINK, just the basic PLINK. It's under Cog Genomics, PLINK 1.9. Any search for PLINK will yield just an enormous amount of documentation on basic filtering, on everything from actually running your own GWAS to just handling different types of genetic variants, a lot of conversions between file types and input files. It's a very rich resource, and it's extremely commonly used, been used for a very long time. So it's a very well-documented place to be. You can find answers to virtually any of your questions on the Cog Genomics PLINK website. Also, for the purposes of this quality control pipeline, there's one package that Hannah Myers has developed, which I've never used all the way through. It looks to be quite useful. It's a R-based program that will perform a lot of the steps that we're going to perform here step by step. It will perform them in an automated sense, a semi-automated sense through R. It takes setting up quite a few reference data sets and files, but it's a very neat run through, and it provides some nice graphical illustrations of your quality control. It's something to keep in mind. There's a lot of very open ongoing development in terms of simplifying and improving quality control pipelines for genetic data, but it's a very messy field. It is a very messy field. I cannot tell you how messy things can get when you are dealing with different genome builds. So this means one SNP will have a certain set of coordinates in one genome build, a second set of coordinates in a different genome build. Every few years, we update our understanding of the human genome, and we actually have to shift things around a little bit. Genes may come and go. Genes may be in new places. SNPs may actually become the same SNP. They may merge as reference data improves. Dealing with different data repositories, formats, different levels of quality of genetic data can become an absolute nightmare. So it's always appreciated when people are working on tools to help simplify these programs. It's fascinating, and some of the things that can be done are really amazing, but there's only a few places in the world where research is being done at the real forefront of handling extremely complex, high-level genetic data because of the complexities involved in just organizing and accessing these types of information. It is extremely large, it's extremely high dimensional, and it's extremely finicky. But I've tried to tailor this workshop in such a way that we won't run into any of the really, really dirty issues, and actually be able to see some of this code just kind of working. So we'll start here with just a quick look at what the actual BIM and FAM files look like. So we can copy and paste that directly into our bash. So this head command will show us the top 10 lines of any file that is a text readable. And the BIM file is just that. It's a text file. It's separated by tabs, tab delimited. The first column is a chromosome. The second column is that RS ID, the identification. The third column is actually a genomic position. It's in a unit called centimorgans. It was how we understood the relationship in terms of kind of distance. It's a pseudo distance, not a physical distance. It's how close two SNPs were thought to be to each other on a chromosome based on how often we observe them together in people and in populations that are identical by descent. So basically, we would have a measure of, where SNP 1 is and where SNP 2 is relative to how often we would find them coexisting as alleles, whether we'd find their alleles coexisting or separated as recombination events during meiosis. That was necessary because we didn't have a full sequence of the human genome, which is what the fourth column is. This is the physical position on the chromosome as to where this SNP is. So this SNP on chromosome 1 is located at base pair position 863,579. And its minor allele is an A, and its other allele is a G. So there we go. We have everything we need to know about this biallelic SNPs. And that's our information for genetic variants. Now, what about the FAM file? Remember, I told you it's also a text file, but it's not giving us information on the genetic variants. It's giving us information on the other aspect of our data, which is the subjects, the actual people that we have. And in this case, we can see the column IDs, which I've also listed out here. A lot of the reasons for this file structure have to do with historic reasons, the way that it was originally designed to deal with family data. So the first column ID is actually called a family ID. So if you were recruited as part of a triplet study, if you had two parents and a child, or if you were all related, you'd all be part of the same family. You'd have the same family ID. And then within that family, each person would have a unique individual ID, which is the second column. Now, in this case, it's a population data set. In fact, part of the assumptions that we're working under and something we'll test for is that no two subjects are actually related to each other. We want to exclude families. So that's why the family ID and the individual ID are the same. Everyone is part of a unique family, and their family ID and individual ID can be interchanged. The other columns have to do with the IDs of mother and father and whether or not we want to put their sex and phenotype value in. We don't actually want to put phenotype or sex data in this genetic file. Again, it's a relic of an age when a lot of genetic analyses were extremely straightforward. You'd have one phenotype, maybe a case control status for something, and you'd embed your sex information directly into the genotype file to perform some quality controls. We don't need to do that. This data set has been sufficiently pre-quality controlled. We know that there aren't sex mismatches. And we also want to exploit the flexibility of the precise tools and virtually every other tool that's been developed since to look at multiple phenotypes and to look at different covariates and use phenotype data that is separate from our genetic data set. So in this case, this FAM file contains only the IDs of our subjects. -9 is PLINK for missing. -9 is handled as missing in PLINK code. So that's it for those file formats. And again, remember the bed file that is the largest of the genotype files is just the binary actual genotype data, which we can't visualize, but is read in by PLINK. So it's 2:10. I think we're going to have a break, which I think actually works out nicely. Double check the schedule to make sure that I'm not mistaken. 2:30 afternoon break. Okay, so you know what? We'll do one or two more things before we go in for the break. Are there any? Can the significance of -9 be explained more? Yes. So -9 tells R that this is or tells PLINK that it's a missing value. So every program, including R or Python, Bash, not really Bash, you don't do too much numerical operation in Bash, but the PLINK program reads in your files, and if a value is -9, it assumes that to be a missing value. So we'll see when PLINK reads this file, it's going to say there's no phenotype data. It's all missing. I strongly dislike that. I think that missing data should be coded as NA or even as blank. But again, it's kind of a quirk of this file format. Interestingly, quite recently, PLINK has come out with PLINK version 2.0 in alpha, and they've generated a new type of file set called PLINK binary 2, which gets around some of these problems, a bit more modern. It can also handle more complex types of genetic variation. It's a little more complex. It's also a little bit more efficient in terms of storage sizes. But this is what we get with a file format that's been around since the beginning. -9 is missing phenotype. And again, the only reason why it's coded as missing here is because I don't care to integrate my phenotype data. So for this sample of subjects, it's 1,000 people, and we have hundreds and hundreds of measurements on these people. And part of a study that's a longitudinal study. We'll discuss more about the data type in this particular sample on Friday when we bring together kind of other types of measurements from these people. But we have tons of phenotype data on these people. I do not want to just kind of pick one at random. It seems to be my most important, stick it in the PLINK file, and it's always there with the genetic data wherever it goes. I like to keep the genotype data separate from phenotype data. Sometimes different phenotypes should be kept separate as well. When you start building up so many different types of data, it's best to keep them separate, but cleanly integratable with the same overlapping subject IDs. So you see, I have subject IDs here, and as long as I know who these subjects are in my phenotype data, in my gene expression data, in my brain imaging data, then I can always integrate down the line. But I don't like mixing it together. So in some cases, it makes sense to fill that in, but less and less these days. Now we'll do the first couple of steps just to verify that PLINK is working before we take our break in about 10 minutes. We're going to perform two types of genetic data quality control. These are the essential ones that you really need to do before you calculate a score. First one is the Snip Qc. So we have to do something at the SNP level for each variant. There are different things that can go wrong in the genotyping process. There are different things that can go awry even at the population genetic level, biologically. There are things we might want, there's certain reasons why we might want to remove a SNP from our data set before we start calculating anything from it. One of those is called Hardy-Weinberg Equilibrium. Now this is different from linkage disequilibrium. It's a different concept. Hardy-Weinberg Equilibrium is it's a formula that dictates the expected random distribution of genotypes in a population where a genotype is not under any type of extreme positive selection. So basically, what this means is that if you have a variant or an allele that is kind of driving or is behind a Mendelian trait or is extremely penetrant for a very deleterious trait or phenotype, or is possibly even lethal in some cases, that variant will start to the distribution of alleles in the population for that variant will start to follow a certain trajectory that pulls it out of the gene pool. Any SNP that is undergoing that selection process, positive or negative selection, will start to deviate from the expected pattern of essentially shuffling of genes that is supposed to occur pretty much randomly every time that a new human is conceived. This shuffling of genetic material during crossing over which occurs during fertilization. So we want to identify any SNPs that might be deviating from this pattern, and they can just kind of stick out as odd, give us very bizarre statistical results because of an odd distribution of allele frequencies. You shouldn't have many SNPs that violate this equation, but it's a simple test to see if the expected distribution of alleles given the allele frequencies are deviating from Hardy-Weinberg equilibrium. We also want to remove any SNPs that have a very low genotyping rate. So this means if we look across all of our subjects and find that there's a SNP that was only genotyped in successfully genotyped in 50% of people, maybe we want to remove it. Maybe it's problematic. Maybe there was something wrong with the oligonucleotide in our genotyping experiment. Maybe there was something wrong with our optical software when we were making the call, or maybe it's just a very low confidence site. Maybe there was just something wrong with the experimental procedure. During imputation, typically we remove entire SNPs that do not have a high quality of imputation. Sometimes in genetic data sets, individual observations will be removed. So rather than just having a full set of genetic data, you may actually end up with more sparse data types where some individuals are missing genotypes for some variants and some variants are missing for some individuals. In our case, that won't be necessary, so genotype rate shouldn't actually be an issue. Low minor allele frequency, we've discussed that a bit, violating the statistical assumptions of our regression, also making it very difficult to successfully derive inference from a distribution of alleles. If you have a very, very rare allele, and even if you're analyzing 10,000 people, or in our case, a sample of 1,000 people, if not even one subject, if it's such a rare variant that only one or two subjects is heterozygous for a variant, then we can't actually make any claim statistically about what it means to have that allele, to have a copy of the allele. Because there simply aren't enough observations with a different genotype to say, well, as you increase the number of C alleles, as you go from 0 to 1 to 2, you see an increase in your risk for liability for MDD. If there aren't enough people in our data set who actually have those alleles, we can't make any type of inference at all about that. There's also the high imputation quality. So I've already done this. I've already retained SNPs that have a high quality of imputation. And then finally, abnormal levels of heterozygosity. So this is basically, and actually heterozygosity rate, this would be done at both. We're going to be performing that at the subject level. So basically, should switch this, shouldn't be in the Snip Qc. This is the best part about GitHub. We'll update it. Once I put that into the subject category, you'll be able to see the updated instructions. Heterozygosity is the idea that generally we will expect, again, having to do with the way that the genome is sorted and recombination occurs in every generation, it's expected that you will only have certain amount of genetic variation at a number of points across your genome. So if you imagine what heterozygosity actually means, right? It means that one of your chromosomes has one version of the SNP, the other chromosome has the other version of the SNP. You have two parents that had different genotypes in one of their chromosomes, and those were the ones that you inherited. Now, in a lot of places throughout the genome, you actually have mostly homozygous genotypes. For very common variants, you are likely to have both of the common variants. Sometimes you might have heterozygosity. If you have a lot of heterozygosity, if we look across 5 million sites in the genome and find that, oh my goodness, you have a ton of heterozygosness, which means that you will, by default, have a lot of rare alleles that you might not expect to find just by chance in the population if you're the product of kind of a random mating procedure. You will see excess heterozygosity in an individual across their genome if they've been the product of inbreeding, where you have rare alleles that may be deleterious that persist in the gene pool because of this inbreeding, because of these consanguineous relationships. You end up keeping deleterious alleles in the population. Or if you actually had a physical contamination of your blood samples for two people or more during the genotyping phase. So when blood samples are being prepared and DNA is being extracted, if a pipette tip or a flask or a tube is is contaminated with DNA from one person and mixed with DNA from another, you're going to observe many more loci, many more SNPs in the genome than you would expect generally and by chance to be heterozygous. Because in places where you would expect people to just have 2 copies of the same allele, you now are increasing the chance of observing a heterozygous read on those oligonucleotides when you mix those samples together and look at the same variant. So we want to remove individuals with abnormal heterozygosity. And we also want to look at a few other things at the subject level rather than SNP level. Again, low genotype rate. Are there any people who maybe had very low quality DNA? Are there any people who were kind of very poorly genotyped or ended up with a lot of low or a lot of missing genotypes after we removed kind of low quality observations after imputation? We also want to check for relatedness. So again, I mentioned some of these file formats are dealing with family data. These are not family analysis. In fact, it's a major confounder if we have individuals who are related to each other. We want to remove any people who are related to each other from the data. We also want to check, and this is something that is actually sometimes more challenging than it seems. We do want to keep in mind that if our data set, right, if the data set we're looking at, let's say we're looking at, again, major depression status, and we aim to generate a polygenic risk score and associate it with depression in our sample to see how predictive it is. If our genetic data, if the genetic data in which we plan on calculating the polygenic risk score in overlaps with the original GWAS in terms of sample subjects, then we will be overfitting our analysis. We will be performing a circular statistical analysis whereby the score that we calculate is based on effect sizes for the very same phenotype in the very same subjects that we actually ran the GWAS on in the first place. This can be challenging when you are analyzing data from large consortia that are participating in major meta-analytical papers. So in the IGAP consortium, in this this International Genotype and Phenotype Consortium for Alzheimer's disease, we have many dozens of samples of people that have contributed data on Alzheimer's disease from around the world. And when those summary statistics are published and we want to analyze them and use them to examine the genetic architecture of Alzheimer's disease in one of our samples, we have to be very careful about what we look at and which phenotype we analyze, because if we try to make a claim about the association of a polygenic score calculated from that IGAP GWAS on Alzheimer's disease in our sample that was actually part of the IGAP GWAS, then we are committing several statistical sins. And everything that we determine from that point will be inflated. So there's those considerations as well to keep in mind. So that's really, I mean, that's pretty much it. The rest of the code, I saw a question that, yes, Aaron responded to. So PLINK and Precise are both installed inside the Docker environment. So when we come back from our break and start running these PLINK commands, we're going to accomplish all of these SNP and subject level quality control steps. They do not take long to run. It will give us lots of information about the genetic data set, about the number of SNPs. You'll get to see it work, you can get a feel for it. And this workflow will be quite effective really for any genetic data set that you might come across in your research to properly clean it for analysis in Precise. And then after the break, we'll go through these quality control steps. We'll run 2 instances of Precise, take a look at some of the output and how you can use it, and then do, if we have time in the end, some kind of preliminary association analyses, reading actual individual level scores into R, merging it with our own phenotype data, and doing some initial exploratory analysis to see if we can identify something in our sample that's associated with a genetic risk for major depression. So we'll pause it there for now. I think that there's another session for day 1, tutorial 2, which is where we will perform the quality control in PLINK and then run the Precise. So if there are no burning questions here for Crowdcast. I think we stick to the Riot Rooms. I'm going to stick one more poll out here for the next couple of minutes. Yes, and thanks for asking, Sunny. Yeah, anyone, this is a good time as well. We'll stop just a little bit early for this break so that everyone has time to talk in rooms. Okay, we've got majority. So anyone who's not quite there, please don't be afraid to speak up in the chat rooms and the gather town. Wonderful. And remember, as soon as this broadcast is over, this is true of every session, morning sessions, as well as the afternoon tutorials. As soon as this broadcast is over, you can pause, you can rewind, you can go back and look at any of these steps. And of course, reach out to us if you have any further questions about that. So we will reconvene we'll reconvene at 2:45 in the final session for the day. We will run the PLINK code, get to see how it processes the data. We'll run precise. We'll take a look at the scores. We'll take a look at the associations. And you will all be much better off than you all were when you woke up this morning when it comes to confidence and knowledge and enthusiasm for the incredible world of multi-scale neuroinformatics, beginning with the genetic core, the genetic foundation of major depressive disorder. So I'll see you in about 20 minutes.